Illinois in chemistry. And as I said, Professor Alex Way was the best chemist I knew in nanorods and plasma and resonant nanostructures. Professor Liu here is the best chemist I know in developing nanotechnology for sensing and imaging DNA. So it's really an interesting connection between the nano world and DNA. So welcome. Thank you.
Thanks, Steve, for inviting me. It's really a great pleasure to be here. And also I'd like to thank Dr. Wei about giving a very nice introduction about the fundamentals of the imaging and all the nanomaterials to make my job much easier in terms of introducing that. So today I'm going to focus on selective agents and for sensing and imaging, and based on the combination of functional DNA and nucleic acid and nanotechnologies. And again, this is I want to thank Dr. Wei again for a very nice introduction. I'm sure also Dr. Bohart have also given a very nice introduction about all the beautiful different imaging techniques, and I don't want to do any further introductions. The bottom line that one of the challenges is how to increase the contrast between the targets that we want to do and the surrounding environment. This is something everyone wants to do because that's giving you more effective imagings. And of course, again, Dr. Wei have done beautifully in terms of talk about different type of material from the shapes and different distances dependence. And those nanomaterials really play a key role in really meeting this challenge of increasing the contrast between the targets and the surrounding environment. And one of the primary applications that has been demonstrated, there are many of them. One of them will be using gold nanoparticles. And this is so-called nanoflare from the Merkin's lab. So basically, they're able to functionalize the DNA on top of the nanoparticle. This is nanospherical particles. In the present different RNA, they're able to demonstrate the increase of the of imaging. So it's called nanoflare. And most of the nanoparticle DNA obviously play a key role, but most DNA nanoparticle prepared after nanoparticle have been prepared. So Dr. Wei have mentioned a lot of different ways of functionalizing nanomaterial by either physical absorption, chemical absorption, and many other different ways. And so what our lab made a small contribution to the field is that what we discovered that instead of making the nanomaterial first and then functionalize later, and then by accident actually the Dr. Zidong Wang actually in the lab discovered that if you synthesize a material, nanomaterial, in this case nanosphere, and in the present different sequences, you actually end up with different shape controls in the present of different sequences. For example, if you do have any DNA, you got a spherical particle. That's not surprising. But instead of using that, using our poly-T, the all T layout together, you also get a spherical particle. But yet, when you start with adenosine or cytosines, you actually get a so-called nano-flower shapes. That's actually a different way of. So what happened then? The DNA absorbed onto the surface of the particle. Depending what sequences it, they either do not absorb very well, you end up with like poly-T cases, spherical particles, or in the poly-A, poly-C cases, it absorbs so strongly, and then it can influence the shape controls. Instead of having spherical particle, you got this kind of a nano flowers type of particle, and then the DNA is a part of the whole growing process. And so that's actually we published in 2000, and then we are warning again. This is only the one completely same sequences, the A in the row or T in the row, and then we're warning about is any even more in-depth detailed information about this kind of controls. And we all know about genetic code, right? So this is very classic basic molecular biology. Depending on different DNA sequences, you can actually code a different amino acid. So this is the foundation of modern biology. And we're wondering about is any such a similar quote-unquote genetic code for non-abiological material like nanomaterials. So based on that particular study, and then Zidong did a little bit further investigations, he basically started out with poly-G, right, or G20 or G30s, and they so slowly changing the different compositions. Again, this is poly-T. This is no longer the spherical particle. We start with the nanoprism, the triangle nanoprism, and then we're adding a small amount of gold, and then slow reducing environment, and then now incubate before the reduction different either poly-G, you give this shape, you got poly-T, you got a different shapes. You got a C, you got a kind of rounded shape, and then poly-A is similarly, but with kind of rough surfaces. So that's similarly to what we shown here. But instead of completely whole one sequences, when we start making, for example, slowly increasing the T's from T10 to G20, T15 to G15, T20 to G30, you can still slowly, you can see migration from this hexagon shapes to this kind of star shapes. And more important, you can go this way too. This is supposed to be A's, right? And same thing here. You can see there's a different composition of different sequences. Now you can get a completely different one. More importantly, we have I don't have time to show you here, this is almost 100% yield. It's not just one thing. If you get this one, almost a whole images give you that particular ones. So we're starting decoding. There could be some kind of a biological, genetic code for a biological material. So that's very exciting to us. And more importantly, because this DNA is part of the growth process, therefore the completely embedded, or not completely, partially embedded into the material is extremely stable. Dr. Wei mentioned earlier about many different ways to functionalize the nanoparticles and nanomaterial, but sometimes it's very hard to maintain the stability. For example, thiol gold chemistry is very popular, but as mentioned earlier, it's subject to oxidation, also replacement by other kind of thiol material, containing material in biological system. In this case, we do not use the thiolate DNA, just complete regular DNA without any thiol modifications. But yet their stability is still maintained. Again, this is poly A, poly C cases. Poly T is not, because it does not give you any kind of functionalization. They have a different interaction with that. Obviously, no DNA can see different kind of stability. That's visually. This is more graphically to show you different kind of extinction of these materials. Another thing also to demonstrate how stable it is, if you do overnight incubation with the mecanoethanol, this is a very popular thiol agent. It can replace 100% of thiol DNA on the ghost nanosphere. Again, same thing if you do that, but with same conditions, but with the nanoflower that we show in here, it's only about 30% replaced. Around 70% still maintained in there, because again, we don't even have thiol on the DNA. If you have no thiol, this is very easily, you don't even need the overnight incubation. It's just right away they can be replaced, because the thiol gold chemistry is much stronger. But yet without even DNA, but this is part of the growth process, they actually very, very stable. So it's really give exciting opportunities to using this kind of new material. It's kind of hybrid material between DNA, part of the nanoparticle, allowed to do some bioimaging applications. So this is preliminary data. We show that nanofluctuated gold particle. This is a nanosphere. This is the kind of the one that I mentioned earlier, the nanoflower kind of a type of material. You do see a very good kind of imagings, but again, when you have a nanoflower, due to again a lot of surface plasma effect, then you can see that a much stronger imaging agent. So it's definitely a good potential application for intracellular or even in vivo imaging on the surface. Yes, go ahead. This one with the cells, it's about 90 minutes. Yeah. And again, we mentioned that there are minimum toxicities. And the cell's still alive. You can see this division, dividing, and so on and so forth. So it's kind of interesting opportunities with that. And also we can show later in my talk about more selective imaging. In fact, that's exactly what my main focus of today's talk, although that result will be shown later. The key word here is selective imaging, because again, many people have demonstrated whenever you make an imaging agent, like nanomaterial-based imaging agent, you want to put a folic acid or RGB peptide, and we want to do a demonstration for that. That's beautiful. And then what happened then when you talk to biologists, they say, that's very nice. You can target something, but what if you can you actually use your imaging materials and technology imaging something I care about? So the key word is the selective imaging, so they can specific targeting to the target they're interested in. So basically, can we detect molecular marker for tumor before that? Obviously, every biology molecular has their own favorite target, right? So this general one you can demonstrate it, but specific even better. Until recently, antibody has been used to provide this selective imaging. You probably, if you go to hospital right now, most likely antibody play a key role in both the diagnostic as well as therapeutic. But however, as you all know, the antibody is quite large. It's huge and also pretty high cost and very low stabilities. Most importantly, actually, from scientific point of view, they're not very effective against small molecular targets. And also target too toxic to reach antibody for. Sometimes you want to do again detections, not necessarily in vivo, but in vitro. For example, sometimes the toxic materials before you. And also There are also a lot of time conditions may not be optimal for antibody because antibody reads at the physiological conditions. A lot of time when you do in vitro diagnostic, a lot of time it may not be, for example, very high organic environment in wastewaters and high pH and low pHs. And also, it's also too big sometimes for dynamic imaging because antibody itself is big and it's a kind of slow diffusions. Sometimes they excel because when you do imaging, you want to be sort of non-invasive. You want to have a bystander to watch for metabolite, for example, floating around. But if you have a big molecule to try to trap it, on the big molecule itself, dynamic can influence that. So it's no longer a true kind of bystander, non-invasive imaging is invasive that way. So there's a lot of interest in this one. So let me just hit home the point about one important applications. Again, we know a lot about how to detect large molecules. We have DNA RNA arrays. We can do the detections. We can use antibody. You can detect proteins. But a lot of small molecular markers are still very important, we all know. But yet we do not know how to detect them or imaging them. For example, metal ions, many organic molecules play key roles, and also many invasive species, toxic chemicals like heavy metal ions, like many organic toxins and so on and so forth. Just drive home the point about the detection of small molecular biomarkers and metabolite. We all know the famous detection of PSA. This is it has been until recently a good considered a good biomarker for prostate cancer, and it's used in the hospital for a long time. But again, this is actually more than 10-year-old studies. This is actually how high a false positive rate, how high a false negative rate. This has been known for more than 10 years, but yet doctors still prescribe this. Fortunately, I think last week there's another news, but about a year ago, there's another one. People already have recommended I forgot the organization, but anyway, it's been recommended not to be used as a way to detect prostate cancer because there's about 50%, this is actually more than that, actually, if you look at the false positive rate. The bottom line, a lot of, they're over-diagnostic and over-treated. So a lot of men actually die with the prostate, the high PSA level, rather than because of it. Right? So but that's really one of the issues. Again, I don't want to get into debate about medical debate, whether it's a good marker or not. But the bottom line, from scientific point of view, it is much more desirable, need to be improved. So in 2009, there's another publication in Nature to show that this small molecular markers has a much better correlation between the onset of the prostate cancer versus that of the high level of this so-called saccharin is a glycine derivative. Again, there's still some debate about this study because it's only a very small population for certain patients, but not necessarily for a whole population. So again, I don't want to get into the medical debate, but the bottom line, I want to show you that the whole field is dominated by protein or large molecule-based detections, and that's not very satisfactory, and that small molecular marker could be, and in many times can be, very good biomarker, but yet we don't know how to find them. But more importantly, we don't know how to detect them, even if we know it's a good biomarker. For example, there are no good detection right now for a detection for a small molecule like this. So the bottom line, a number of non-DNA RNA protein biomarkers remain to be discovered first, but more importantly, remain to be detected and imaged. So correlation of these small molecular marker metabolite with large molecules in the level, but more importantly, the biological function or lack of it, is really an unmet challenges in molecular imaging. So that's the focus of our work in today's talk. So this is easy said than done because when you detect DNA, you know based on Watson-Crick space pair, you can detect corresponding DNA to bind another complementary DNA or RNA. When you have a protein, you just inject animal with whatever that protein called antigen, you can raise antibody for. But for small molecular marker, as I mentioned earlier, antibody is extremely poor at detecting small molecules because if you elicit immune responses, it's extremely difficult to do that. So it's not very readily. So it's not people do not want to detect small molecules, but just because people do not have a general method to be able to, first of all, obtain the molecule to bind any specific target. Not only heavy metal ions, small molecules, but also other type of different kind of molecular markers. But also selectivity is a key thing because those small molecules are very subtle. And the functional group is very limited and very subtle. Sometimes one from hydrogen to with the with OH, or one small methyl group versus the ethyl group can make a big differences in their functions. But yet, how do you have a molecule to be able to differentiate between the two? That's also very challenging in terms of improved selectivities. And that's not alone, but also having a molecule bind to another molecule, that's not necessarily a success until you can be able to image it. So we have to have a signal transduction. So how do you transform the molecular binding or recognition event into something one can detect using the very beautiful imaging technique we developed? That's also a challenge in a general way. And finally, the dynamic range, because every single metabolite has only certain threshold below which is called the normal, and above which it can be having causing diseases. So so-called threshold levels. Having a good imaging agent and it's not good enough if you only detect either way above or way below that level. And for most people, really what people care about is detecting, having imaging changes most dramatically around the threshold levels. So how to tune that kind of a so-called dynamic range, so the matching that is also quite difficult. Again, a lot of success has been published, demonstrated in individual cases. But very few methods generally enough, like antibody cases where you have a general method. Once you know how to do one, you can actually apply to many other ones. So that's really huge challenges for detecting, so target biomarker in general, but specifically, particularly for small molecular markers. So that's really what we're focusing on. So first of all, can you actually have a general method? On one hand, you can detect pHs, you can detect viruses. At the same time, you can actually apply the same method for metal ion, toxin, organic cofactors, and bacteria, and so on and so forth. And so that's really the key. Again, as mentioned earlier, until recently, antibody has been pretty general. That's why it's been dominating the whole diagnostic imaging market. But as mentioned earlier, it's not very good detecting small molecular markers. How do we overcome that? So let's come to today's talk, basically functional DNA. So what's the DNA? We all know this is the DNA, and we all know the RNA. The difference between the DNA and RNA is basically one is H and OH group in between the two. And so we all know DNA is a good genetic storage material. That's why we're different from each other. And RNA is a genetic transfer information material. But in the early 1990s, in the Wire-Windler discovery by two different groups, that showed that RNA is no longer just a genetic transfer information material. It actually can be enzymes called ribozymes. Most of you already know that won the Nobel Prize. Basically, they can have enzymatic functions just like protein enzymes. But in the early 1990s, in 1994, actually, Breaker and Joyce have demonstrated that the DNA, just like RNA and proteins, it can be enzyme too. It's called DNA zyme. So basically, it's one kind of piece of DNA bound to another piece of DNA in the presence of cofactors. It can have a specific enzymatic reactions, all exclusively using DNA as an enzyme functions. About the same time, 1990s, it was shown that they're called the aptamers consist either DNA or RNA that can bind specifically to a target like antibodies, very selectively. You can either combine the DNAs and the aptamers in called aptazymes, basically a piece of aptamer and couple with the DNA zymes. And so they show allosteric effect in the present this one, they can either enhance the activity or inhibit the activities. So the combination of DNAs and aptamer and aptazymes is collectively called function DNA. Why do we call it function DNA? It doesn't mean that regular DNA does not have function. Of course it's a function, but they have a function beyond the genetic storage information materials. It can be enzymes, it can be antibody, or combination of both. So that's really what DNAs and function DNA do. Also similarly function RNA. We prefer functional DNA because functional DNA is much more stable and much cheaper to produce. So that's what I mentioned earlier. The beautiful thing about functional nucleic acid that you can have this very powerful but yet quite simple method of called combinatorial selection or in vitro selection of selects. This is the acronym for system involution of ligand by exponential enrichment. It's basically, the idea is quite simple but extremely powerful. So basically what happens, you start with a large DNA sequences, with a library of many different sequences, up to 10 to the 15 different sequences. That's about 100 trillion different sequences possibilities. Among them, that we want to know there's maybe one sequence is capable of binding to a specific target, and it's covered of certain functions. And the nice thing about this method, then you can do selective amplification. You basically, for example, you can immobilize some kind of metabolite onto a column. There are many different ways to do that. This is one common way to do that. And they ask a simple question: among the 100 trillion different sequences, is any one or few sequences capable of binding to the target selectively and strongly? And then you can actually separate them out from the column chromatography, and they can amplify by PCR. That's another beautiful thing about nucleic acid versus other kind of method like organic molecule-based or protein-based. You can use the PCR to amplify a single molecule into many different classes. So when you do that, you can do the selective amplification, and you can also introduce a mutation. And so you can actually, this is mimicking natural evolutions. So they can actually enrich that particular sequences. You can actually go back to do the further selection and have more stringencies. Go ahead.
So you select them by having a substrate which they bind to, and if they bind to it, then those are the ones you select on?
Or you can call substrate, or you can call cofactors. Basically, whatever target you want, and whatever reaction you do. Yeah.
How do you select the specificity?
I'll show you that in a couple slides. Yeah. So basically, you supply the target of interest, and then you can do the selection. You amplify that, and then you also, you take about a few days, depending how good graduates and how hard they work, obviously, too. And normally about 2 days per run. Good graduates can actually do 1 day per run. In fact, Ron Breaker, this is the person, the postdoc, when he enjoys life, he do 2 runs per day. He told me when he did that. So most people do 2 days per run. He did 2 runs per day. Okay. But anyway, so that's the average student. So, and it's about a few dollars per run, and then before you can do many different runs. The bottom line, you can reach to a satisfaction kind of affinity or concentration you want. So this is a kind of demonstration done in the early 90s by 3 different groups, pioneered by 3 different groups, and now being utilized by many groups now, including my own lab. This is a summary about, this is almost many years old now, but the bottom line that this actually has been enriching every day. There are online databases to have a kind of list of targets that have been recognized. The bottom line that it can recognize both larger molecules like antibodies, but also smaller molecules. That's what I want to focus on. And a lot of groups actually focus on trying to replace antibodies with aptamers. And what we actually believe that actually we can actually do even better by actually finding out the gaps in the antibody technologies. What we believe these are the gaps. For example, you don't find many antibody for metal ions or many small organic molecules because they're not very effective for them. So that's what we've been focusing on. This is a summary of the published work, not the proposed work for both small molecules and large molecules. And just give you a flavor about the type of thing that we have done, and to show you how effective they are. For example, for DNA that bind different metal. For example, this one is very effective selectively for magnesium. And this one very selective for these sequences for lead, this one for copper, this one for uranium, and so on and so forth. And so that's really what gives you selectivity for that. And then they all have a general structure. This will become important when we talk about turn into a single transduction form. So that's what I hope to show you. There's an alternative method from antibody to do the selection. It's quite general. For example, we can have a large DNA library. You can ask a simple question about which one of the sequences bind to a specific target. So for that one. But then back to the selectivity issue. How they can improve selectivity, right? So this is what most people do. You have a very good intention. You have a method, either using organic molecule, protein, peptide, you name it. You can actually, this is what everyone want to do. They only want to focus on the target, right? So that's really what they will do. But what end up most people doing is actually bind someone else. What happened then when you do a survey of the particular one you obtained, it actually turned out to not only bind to the target you want, but also even bind something else even better. So what do people do? They have to publish, they have graduates and have to graduate, and the professor to get the funding. What they do, this is what the typical thing. Rather than shooting at a dart, aim at a target, they shoot somewhere, and then they draw a circle around it. Okay, I don't have the target, I don't have a sensor or imaging agent for target A. I tried it, it didn't work, but find something about even something else, even B. So that's really very commonly occurring in the sensing imaging world. And that whatever the imaging they can do, that's what they do. So as I mentioned earlier, this is not satisfying for really want to make a general method, right? It works for certain things, but a lot of actually imaging sensing agent is by accident. They just find out they find something else, not intendedly to. So again, back to how do you make sure this one won't occur? In fact, this also occurring, this kind of a problem even occur in mutual selections. I think that's what your question alluded to. So this is what we intended to do. We got a grant from NIH. we want to select the DNA that bind to the cobalt, right? So we did the whole selection. This happens most of the time that what we select out does bind our target, but there's no guarantee it only bind our target strongly. In fact, if you look at a survey, there's the two other targets that bind even stronger, right? Zinc and lead. Okay? So this actually occur quite often too. So how do you prevent that? How do you make sure it only binds to your specific target, but not something else? So that's what we actually decided to do using the negative selection strategy. So what happens, the nice thing about the mutual selection, we do not start with the one molecule and try it, it didn't work, and then go back to the drawing board and do another molecule and try it again and again and again. This is many trial and error processes. So what happens we have a large DNA library. Even though we start with a small subset, very small percent of whole large library, but because our library is huge, we still do not end up with a one type of sequences. We actually end up still with thousands of sequences. So what we do then, if we continue to do positive selection, we have a DNA that binds only to not only a target but also something else. That's why I give it low selectivity. But what we can do then, we can actually adding this competing target to remove the sequences that bind to the competing target. So in principle then, the remaining population should be able to bind mainly, if not only, to the target, because we can remove the population. So then we'll end up with not only high activity, but also high selectivity. It's quite reasonably general, is that as long as we know what the competing target is. For example, we can use the cell extract or serum as a competing target. We spike in whatever target we are interested in. So we can remove the sequence only bind non-specifically to anything in the cell extract, but only using the negative selection, so the population only do something plus your target. So that's basically the concept. This is the result of one negative round of selection. You can see the increase of the target and then decrease of that. So that's how I hope I answer your question to improve the selectivity. Okay? So what about this one? Again, this actually turned out to be not trivial. You know, a lot of time you can have a molecule. In fact, a lot of biomolecules bind to the target very well, but they're not called imaging agent or sensing agent. Why? Because not only you need to have a molecular recognition part, but also have a signal transduction part. This is dear to everyone's heart in here, right? You need to see it. You know it's bind, but people use P32 to labeling to see it bind using gel shift, but that's not imaging, right? You don't want to do, so the imaging, they have to be able to see it dynamically, sensitively. So this signal transduction is really a big problem, particularly big problem for small molecules. For large protein molecule antibody, people do the sandwich assays very easy because they have secondary, primary antibody, secondary, and so on and so forth. You can do the reaction. For small molecule, you do not have that many repertoires for different type of recognitions. How do you do that? So before our work, this has been considered a general method called replacement technology. You put some kind of reporter group, either chromophores or fluorophore or electrochemical agent, so that in the absence of the target, this one sits at the binding pocket, okay? So they have a one environment that give you one color or fluorescent or electrochemical responses. So then what happens when the target come in here, all right, it kind of bind to it. Sorry. Oh. The idea is this. So basically when it bind to it, it replaces that reporting group, okay? So that you have a change of color or fluorescent signal or electrochemical signal. So it's supposed to be very general, but the problem is the following. For large molecules, this may be okay, but for small molecules, remember these reporter groups are no longer small. This is actually exaggeration because a lot of reporter groups are large aromatic organic molecules. So when you put in there, that's not what occurs most of the time. What most of the time occurring, this guy is just as big, sometimes even bigger than small molecule like a metal ions. So it's actually changed the character of the binding site. You spend several months or weeks to get something to bind to your target, but when you put your fluorophore in here, you change the character of the binding site completely. So when you do that, then you lose the selectivity. It's no longer bind selective to your target. So all your effort in making selecting agent for that signal transduction does not occur because of that. But more importantly, a lot of time you don't even know where the binding site is. For example, if you look at the table, there's less than 1% of those kind of DNA or aptamers have a crystal structure. Without three-dimensional structure, you don't even know where to put your reporter group in there. So this is not even, you don't even know, it's kind of a blind in front of the elephant. So that's really one of the big challenges. So therefore, if you remember, it's 1990s, this kind of method had been known. But the kind of sensing imaging activity has not been going on for more than 10 years because of kind of this challenge. There are some successes. People did succeeded in doing some small one target, but they cannot be applied to another one because every single binding pocket is different. Where you put it and how close to do it is very difficult. So what people do then, let's put away from it, right? That's easy. Let's make away. But the problem is when it bind, have nothing to do with where the signal transduction occur, right? So then what you do is become trial and error process is you have to put something closer, but not too close. So again, as I mentioned, you don't even know where to put it because 90% of them do not even have a crystal structure or three-dimensional structure. Even if you do have it, you have to be careful not to be too close or not too far away. So it's become a trial and error process. So what we decided to do is that we're not going to repeat this one because it's going to be too hard and not very general. We take advantage of very simple, again, very powerful concept of melting temperatures. Because if you look at this one, they all have a very similar secondary structure. Why? Because we use the same kind of techniques. Start with the same DRAM library. The only difference is the different sequences in here. These sequences give you selective for magnesium. This one gives selective for lead, and so on and so forth. And the nice thing about that, they all have a similar secondary structure. Then they all have different kind of melting temperatures. So what we did then, we play a simple trick of melting temperatures so that when you have a different DNA hybridize it, this is a complete hybridizing here. This is the one that's very selective for lead. And this is blue kind of funded bases are conserved sequences for lead selectivity. But this one has a different hybridizations. So what we can do, we can carefully choosing the number base pairs as well as the GC content. So we can actually make this one to be completely hybridized at room temperature, or any temperature you want, but room temperature obviously is when people do detections. So then we put a fluorophore here and quencher there. So this is schematically, we put a top strand with a fluorophore like that, and the bottom one with a quencher like that. So when they hybridize together in the absent target, they'll be quenched. The fluorescence of the signal will be quenched by the quenchers, so they'll be hybridized together. Have a very low fluorescence signal. But then when target come in, they have either two things. One is the binding, but more importantly in this case, it's the enzyme that can cleave. When they cleave, they can actually change the melting temperature from the whole thing here into half of it. So that we can actually make this one to be short enough so that it will be dehybridized because the melting temperature will be below room temperature. So what happened then, it will not stay hybridized, it will be released. When they release, the fluorescence signal should increase. Why is this general? Because remember here, as long as we know the sequence, this one doesn't matter. As long as you have any kind of hybridization, it's not important for selectivity. Important selectivities are in here. So the bottom line that when you have one the basics, the general method, you can allow it. This is the concept. This is a proof of concept. We published about 12 years ago. This is the whole thing by itself, right? Have a very high by itself. Our sensor, or any sensor actually, you can only detect the so-called mobile targets, something that's floating around that can be kinetically and grab onto it. Something that really so tightly will not release it, we will not see it. So in other words, we cannot detect total lead. We can only detect the lead that's mobile.
Can you please go back to the slide where you said you saw the way you did experiments with your selective?
Oh. Yep, this one?
Okay, so I still don't understand the experiment. So the MCF7,
We incubate the aptamer, the selective binding to the breast cancer cell, MC7. The aptamer selectively bind to that in turn. This is actually the picture. They can actually selectively do internalization called EPR effect. So they can internalize it and then dump the load. So we encapsulate both the cisplatin inside the liposome as well as fluorescing agent, near IR fluorescing agent. We can see both fluorescing agents. At the same time, we can see selective killing due to the presence of cisplatin cancer, anti-cancer drugs.
okay, and then your prostate cancer cell?
Because this one, the prostate cancer cell does not have this aptamer. This particular aptamer do not recognize the surface receptor on the prostate cancer cell. Therefore, there's no selective internalization, there's no killing in here. Because the selectivity due to the aptamer. Because the difference between both, they're both cancer cells, right? But then their surface receptors on the cancer cell, the breast cancer cell has a different expression than the prostate cancer cell. That's how selective this aptamer is. A lot of times there's a more general, like folate is more general for any cancer cell. But this one is additional to that. It can actually do more selectively. It will not touch, it will not recognize other type of cell, only the breast cancer cell.
Will it not also recognize normal breast cells?
Excuse me?
Can it also recognize normal breast cells, or it will not?
No breast cell. Of course, yeah. We have another control. I want to show you how selective they are. So most of the time, most of the drugs, most of the selective agent only recognize between the cancer cell or the regular cell. But in addition, we can do that. We can also differentiate different type of cancer cells. Obviously, yeah, the regular cancer cell. Let me see. This one actually is a regular cell. Okay, but this is folate, so that's not actually the white imaging. But yeah, so our aptamer, it will not touch any kind of regular cell. There are no toxicity issues.
So you kind of demonstrated that like you can engineer these things to bind to specific compounds or whatever. So can you make it so that you kind of have like these Boolean operators where it has to say bind to two things to be activated or bind to one but not another?
Yep, I know what you get at. So in fact, we have in the same paper we did that, exactly that. So this one you can see the either or all, right? So E is one, all the other, or both. Right. So that's kind of a can add to. We have another paper to do this kind of all or AND gate. This is exactly what you're talking over again. Yeah.
What about a non-Boolean approach where you can design, I guess, something like your DNA enzymes with the kinetics? So if you fit it to a different time point based on the affinity, you can determine what.
Yep, and that's not our lab focus because we're not, there's actually a collaboration between mathematician and DNA biology lab. There's several publications that done exactly what you did. They basically just play the rules of two different DNA enzymes. They can do one inhibition, right? Not, or they can do all in all different times. There's actually several publications that does that. Yeah, and that has been done. Not in my lab, but in other labs.
What instrument do you use to measure the fluorescence? I'm just wondering what levels the next.
Right. So the most of the publication done like this is using a laboratory equipment, big fluorometers. But I don't think I have a slide for it. But there is actually a local startup company who licensed the technology from my lab. They actually have a portable fluorometer. They actually can do it 4 different type of metal line right now on commercially, but they also have other coming up. It's a small portable fluorometer. You can do in 2 minutes to do the detection of lead, mercury, uranium, and copper in 2 minutes at the EPA level. So for them, all they care mostly about what's the threshold defined by EPA. For 4 metal line, they can detect this one at the threshold of the EPA level, or slightly below that, because they need to give a quantitative numbers. So portable fluorimeter is able to do that. And I mentioned earlier already, the detection limit slides is somewhere here. Right, so this is the threshold detection level 4. This is the EPA maximum contamination level, 75 nanomolar for lead, 126 nanomolar for uranium. This is probably the lowest, mercury, 10. So these are our thresholds. We can detect below that.
Do you find that, those are 2 questions that are probably connected, do you find that the sensitivity and specificity that you can achieve with this method sort of decreases as the target becomes more complex?
Absolutely, yeah.
And is it possible, I saw on the slides that you developed one to detect GP120, is that sort of preventing you from using it as like a viral detection tool or diagnosis device?
So the detection limit suffers when you have a more complex medium. So that's why we want to be way below the detection limit. Many of them actually about 100-fold or at least 10-fold in there. So that we do suffer by an order magnitude, but still better, good enough to do get a level for that. But more importantly for lead, for example, the threshold for lead is 75 nanomolar for lead in water, but also lead in blood is actually higher too.
What about like viral, like I saw one of the slides you were
Mhm.
detecting the G4 protein, viral protein?
Right.
Is it possible to detect that in anything relevant to small concentration?
For that particular one, depending on the target, depending on what defined by the medical field. Some of them we can do well, some of them we don't, because sometimes you really require to detect single wire viruses. Normally, we cannot do that with a portable one. For the very fancy equipment we have in Beckman here, we may be able to achieve that, but for the portable one, no, we cannot do that. It's really dependent on the target.
Okay, let's thank Professor Lu.